Welcome to today's lesson. Today we are looking at paper 3 2009, and let's see what you are supposed to do. In question 1, you are provided with the bones, bone K, bone L, M, and N and P. Then you are told in Roman 1, identify the bones and name part of the mammalian body where each was obtained. First of all, examine the bones carefully and see which part of the body they are from. Now, in bone K, as you can see, this is the humerus. This is the humerus, and bone L, this is the scapula, and bone M, this is the femur. This is the femur. And then bone N, this is the tibia. This is the tibia. And then bone P is the ulna and radius. Now let's answer our question. Identify the bone and where it is found. Now bone K, we have said this is the humerus. The humerus, bone K. Bone K is the humerus, and it is found in the upper forelimb. Upper forelimb. While bone L, scapula. Bone N is scapula, and it is found in the shoulder region. Shoulder region. Then bone M, I've seen that is the femur, and then it is found in the upper forelimb. No, sorry, the upper hind limb. The arm is known as the forelimb, while the leg the hind limb. Now the N was the tibia. Now this is found in the lower hind limb. Lower hind limb. And then P is the ulna and radius, and it is found in the lower forelimb. Lower forelimb. Yeah. Then you are told, name the parts labeled 1, 2, 3, 4, and 5 from the bones. Let's see those parts. Now, as you can see, this is the number 1, number 2, number 3, number 4, and number 5. Now the first one is which it is found in the humerus, number 1. They are the lateral and medial condyles. The condyles, or you can simply call them condyles. And then number 2 is found in the scapula. This is the glenoid cavity. Yeah, number 3 is found in the femur, and this is the head of the femur. This is the head of the femur. Then number 4, it is found also in the femur. The femur, and it is here. This one. That one is the patella. The groove of the patella groove. It is found in the knee there. And then number 5, this is the shaft. This is the shaft. Now we can label our parts. Can label the first one we have said. Yeah, Now we can name our parts. The first one, you have said the condyles, or the lateral and the medial condyles. And then the second one, you have said, is the glenoid cavity. The glenoid cavity. Third one is the head of the femur, the head. Then the fourth one, the patellar groove. And then last one is the shafts. Then you are told, name the bones that form a joint with bone K at the anterior and the posterior, and in each name the type of joint they form. First of all, you should be able to understand these terms, the anterior and the posterior. The earlier video we said that anterior is the upper side, while posterior, this is the lower side. Now when you have identified like that, now you look for the bone K. Bone K, this is the humerus. And then you should be able to know at the upper side which joint they form. Bone K, they join together with the scapula. The humerus at the anterior, they join with the scapula and they form a ball and socket joint. And then posterior end, they form a joint with the ulna and radius, and then they form a joint known as the hinge joint. Then the next question, we are being told, state the function of the structure labeled 6 in bone P. Now you look at the bone P. This is the ulna and radius. And then part 6, number 6, this is the olecranon process. Then you should be able to know the function of the olecranon process. The function is to prevent overstretching of the forearm or the forelimb. Also it offers a surface area for muscle attachment. You can write like that. Also, the limits the movement of ulna and radius.
I hope you are enjoying the lesson. Do me a favor and subscribe. Thank you.
Now we go to question 2. You are provided with substance labeled P, Q, X, Y, and Z. P and Q are the food substances, while X is the dilute hydrochloric acid, and Y is dilute sodium hydrogen carbonate, and Z is the Benedict solution. Carry out the test to determine the food substance P and Q. Now here you should be able to know the reagents. Now, like if for example we have X, Y, and Z, that is HCl, sodium hydrogen carbonate, and the Benedict solution. You should be able to know which food substance the 3 tests. Now these tests for non-reducing sugar and reducing sugar. Also Benedict solution tests for reducing sugar. Now you have a clue that we are testing the non-reducing sugar and reducing sugar using food substance P and Q. Now here, because our space is very small, let's write here. Let's use this space You write now the food you are testing for, the food substance now, and then you write the test, and then the procedure to be followed, procedure, observation, and finally the conclusion. And now here, then you rule. Then you start with your food substance. Let's start with food substance P. We test for the reducing sugars. Now here the procedure is put 2 milliliter of solution P in a test tube. Add equal amount of 2 milliliter of Benedict's solution, and then you heat to boil. Now your observation will be the color changes. The color changes to orange. Now here the reducing sugar are present. Reducing sugar are present. Then you rule. Now we are finished with the reducing sugar with food substance P. Now here you should be able to identify the marks first. And you have seen there are 12 marks, and you are testing for 2 food substance. You should test one substance, 2 food substance. Like here we are testing now. Now here we are using now. Let's go to use Q. We test also for the reducing sugar. Then we repeat our procedure. Put 2 ml of solution Q in a clean test tube. And then you add equal amount of the Benedict solution. Equal amount of the Benedict solution. Then you heat to boil. And then here the blue color of the Benedict remains blue. The blue color of Benedict's solution remains, or retains color, remains blue. Now here in Q the reducing sugars are absent. Sugars are absent. Now up to here we have good 6 marks. Now because we have seen we have the 8 good marks. Now we have seen we are testing. Now we test for the non-reducing sugar using food substance Q. Now we write non-reducing sugar. Then you say put 2 milliliter of substance P in a test tube. Then you add 3 drops of dilute HCl and then you boil for 3 minutes. Boil for 3 minutes. Then after you boil, remove and cool with cold water. Then after you have cooled, you add sodium hydrogen carbonate. Add sodium hydrogen carbonate dropwise or drop by drop until fizzing stops. Then you add 2 milliliters of Benedict solution. Then you heat to boil. Now you should observe the color changes from blue, green goes there, yellow, and then to orange, the last color. Then you see none reducing sugars, they are present. Then you get your 12 good marks. Also, you can test the non-reducing sugar using P. Then you will get your good 12 marks. Then question 3. The photographs labeled W, X, Y, and Z show seedlings that were grown under different conditions. We told to examine them. Then you can examine them carefully. Then you are told label any 3 parts of the seedling in part W. Now you should be able to know the parts of the plant. This is a bean. You should be able to label them. Then Roman 2, you are told name the type of this was the type of germination, not generation, germination exhibited by the seedlings. Now this is the epigil germination. The epigil germination. Then you're told give the reason for your answer. You say the cotyledon emerge above the ground. Emerges above the ground. You are told the seedlings in photographs W and X were planted at the same time. W and X you have seen were planted in the same time. Then state the condition under which the seedlings were grown. Let's see those seedlings. As you can see, this one is very tall while this one is very short and big stem. Now this was there was absence of light because it is very tall, and this one in the presence of light. Now your answers will be seedling in photograph W was planted in a dark, grown in a dark, while in X in light was grown in light. Then when plants grow in condition named for seedlings in photograph W, they exhibit a certain phenomenon. Name the phenomenon. The phenomenon is when you see the seedlings that are planted in the dark, they exhibit a phenomenon known as the etiolation. And this helps for the faster growth to reach the sunlight. Helps for the faster growth in order to reach the sunlight. Then you are told, using observable features, state only 3 differences between W and X. The W and the X. I will say X has broad leaves, while W has small leaves. W has small leaves, and then X has big stem, while W has thin stem. Also, X is short while W is long. X is short while W is long. Then you are told, next question, seedlings in photograph Y and Z were planted at the same time but under different conditions. Explain how the response exhibited by the seedlings in photograph Z occurred. Let's see those seedlings. They are here now, Y and Z. Now we are asked this phenomenon in Z. Now this is simply the phototrophism, the auxins, the relationship auxins and the phototropism. Now you should be able to describe the process. Now here you'll say, when seedlings being subjected to unidirectional light, they cause auxins to migrate to the dark side, of the shoot tip, Therefore, higher auxin concentration on the dark side, higher auxins concentration on the dark side, stimulates, and there are many, they stimulates rapid cell elongation on the dark side than on the light side. On the dark side than on the light side. This results into bending of the shoot tip towards the light source. Thank you. Now welcome to another lesson. Then today we are tackling the 2010 paper 3 biology, and let's see what the students were being asked. In question 1, you are provided with a whisking tubing solution labeled L, iodine solution labeled solution E, and the Benedict solution labeled as solution F in the piece of thread. Tie one end of the whisking tubing tightly using the thread provided. With the help of the syringe, put 10 milliliter of the solution labeled L into the whisking tubing and then you tie the other end of the whisking tubing tightly. Then ensure that there is no leakage at the both ends of the whisking tubing. And then you wash the outside of the whisking tubing with water. Place the whisking tubing upright in 100 ml beaker. Add distilled water in the beaker to reach the level of the liquid in the whisking tubing. Then you allow it to set up and stand for 30 minutes or more. Now here, when you see the whisking tubing, this is concept of osmosis. That's the concept that we are testing here. And then you are told in A, using 2 mm in a test tube in each, test for the food substance in liquid outside the whisking tubing. Using test 1, iodine solution E. Now here the procedure will be very easy. You will only add 3 drops of the iodine solution, and then you shake. Add 3 drops of iodine solution, and then you shake. Here the brown color of the iodine will be retained. The brown color of iodine is retained. Therefore the starch was absent outside the whisking tube. Starch was absent. And then in the Benedict F, in solution F, now you put the 2 milliliter of the Benedict solution in a test tube, and then you heat to boil. Then the color will change to orange. The color changes to orange. Here the reducing sugar are present. Then in B are told, using 2 ml, the test tube in each case, tell the food substance that was the food substance inside. Now here was the outside, now here is the inside. You are using the same test. Now here you also add the 3 drops of iodine, and then you shake. Here blue-black color is observed. Then here the starch are present. Then Roman 2, the procedure is you add 2 mm, you repeat the procedure, 2 ml of Benedict, and then you heat to boil. Then the color changes to orange. Therefore also here the reducing sugar are present. And by now our students should be able to know these all procedures to test for any food substance. Now you are told account the result in A and B. Here now, as you have seen, outside the whisking tubing, the starch molecules didn't pass, therefore the starch was absent, and inside the starch are present. Therefore you will say the whisking tubing allowed the whisking is very semi-permeable because it allowed the small molecules because more permeable, because it allowed small sized reducing sugar to pass inside. Reducing sugar to pass, so you have seen reducing sugar was present to pass. Why? But didn't allow the starch molecules to pass, because they are large starch molecules to pass, because they are large. That's all. Now question 2, photograph labeled JK, M1 and M2 a section of the mammalian heart. Examine them. So you can see these are the hearts, then you are seeing the blood vessels there, and then you are told in A, the blue, green and the cream string go through various blood vessels and end up various chambers of the heart. For each string, name the chamber where its string ends and the blood vessel through which strings goes. Now for our photograph, they are not very well visible, but the blue one was from the right ventricle, while the green one was the left ventricle, and then the cream and cream 1 and cream 2 was the right auricle and cream 2 the left auricle. Now in the heart you should be able to know which is the right, which is the left, which is the ventricle, which is the auricle, like that. Now let us answer our question. I've said the blue one was the right ventricle, and then here the green one was the left ventricle, here was the right auricle, while the second one was the left auricle. And after you have identified those parts, and then you should be able to know the blood vessels around those regions. And then here, like right ventricle up, there is the blood vessels known as pulmonary artery. Even here left ventricle, the aorta is there, the branch of aorta is there. And then right ventricle, the vena cava is there, and then left the pulmonary vein. I'll advise a student to go to book 2, transport in animals, and then look the structure of the heart very well. And then you are told to name the part labeled 3 in K, photograph K. Now 3 is here, this one. This is the septum. That is the septum. So number 3, this is the septum. And then you are told in C, the parts labeled 4 and 5 are walls of the 2 chambers of the heart. Account the difference, their thickness of the walls. The 3 and the 4 are the 4 and the 5. So you can see 4 this is this one is very thick than this one. Why? Because this pumps blood all parts of the body. That's the reason for the thickness. You see 4 is thicker than 5 because 4 pumps blood for long distance. or to all parts of the body. Then you're told with a reason we should identify the type and each blood vessels X and Y. These are the X and this is the Y. The X is I think this is the vein. That is the vein and this is the artery. You should identify this is the veins. Then we are told with a reason to know why is it a vein. How do you know this? Because it has a thin wall, has thin walls. And the other one is the artery. This opposite has thick wall, as you can see. These are thick, thin, has thick walls. Then you go to question 3. The photographs labeled Q, R, S, and T are sections of some of the plants. This is the bean pod, the orange, and this is the tomato. And then the next page we have the mango. Now you're being told name the type of placentation in specimen shown Q, R, and S. This is the Q, R, and S. This has a margin of placentation because these seeds are found in the margin. And then photograph R is the axial placentation. Also here all the central placentation. Also here this is the axial placentation or the central placentation. Now you answer your question. The first one we have seen, this is the marginal placentation. The orange has the axial placentation, and this is the central or the axial placentation. Then B, label a seed in photograph R and S. R and S. As you can see, my R is the orange, and this is the seed, while this one for tomato, this is the seed. Then you continue. Then you are told name the parts labeled 6, 7, 8, and 9, and 10. This is from the mango. These numbers. Photograph T. Now my number 6 is the outer part. This is the epi cup. And then my number 7, this is the seed. Number 8, this is the endocup. And then this my number 9, this is the mesocarp. And my number 10, this is the remain of the stalk, remain of the flower stalk. Now we label them in the answers. I've said number 6, the epicarp, seed, endocarp, mesocarp, the stalk, the remain of the stalk, remain. Then I'm told give a reason in each case and name one mode of the disperse of each specimen in photograph Q and T. Q and T. Q and T. Q is the bean pod, and then T, the T as you can see is the mango. Q now, the mode of dispersal is the self dispersal. Why? Because the bean pod has lines of weakness. all the searchers. Then T, that the animal dispersal, the mango. Animal dispersal. Why? Why have we said the animal dispersal? Because as the fleshy mesocarp has fleshy mesocarp. That's all. Thank you. And to all my viewers, I appreciate. Continue viewing and sharing and commenting. God bless you.
I hope you are enjoying the lesson. Do me a favor and subscribe. Thank you.